Hi, everyone. Thanks for tuning into this episode of Cell and Gene, the podcast. I'm Erin Harris, and I'm joined by Dr. Michael Severino, CEO of Tessera Therapeutics, the Somerville, Massachusetts-based biotech that's pioneering a new approach to genome engineering through the development of its gene writing and delivery platforms. We'll take a deep dive into all of that for you, but first, Mike, welcome. Thanks for your time.
Pleasure to be here and very nice speaking with you, Erin.
Absolutely. Okay, so I gave a very brief overview of Tessera, and I would like for you to really, um, you know, explain who we are, your mission, and what you're working on.
Certainly. So Tessera fundamentally is developing a platform that we call gene writing. And we talk about the space a little bit differently than others, and that's intentional. And it's not just because we're trying to brand ourselves or we want to find a term that we can trademark. It's because we think about the space differently. In the field of genetic medicine, which is really just in its infancy, today people tend to define the therapies and the companies based on the tools that they use. So if you use one set of tools, perhaps CRISPR-based systems to knock out genes, you're a gene editor. If you use viral vectors, principally AAV vectors today, to deliver transgenes, you're a gene therapy company. If you take cells out of the body and perform complex manipulations, because that's the only place today where you can do that, and then put them back in the body, you're a cell-based therapy company. But underlying all of those approaches is basically the same fundamental intent: to deliver new instructions through genetic code to cells to either restore function or to impart new capabilities like cancer fighting capabilities. And so when we founded Tessera, we set out to build what we believe will be the definitive genomic toolkit. So we didn't want to be limited by, the first generation of a tool that we had in our hands. We didn't want to grab a hammer and run around looking for things that look like nails to hit. And sometimes even if they don't look like nails, you hit them anyway because you can't do anything else if you're limited by your tool. We wanted to build that definitive toolkit. So we wanted to make virtually any change in the human genome that was needed to address human disease. And that led us to the study of mobile genetic elements and the gene writing platform that we're pursuing today. The second essential part of Tessera is delivery, because we want to make breakthrough medicines. It's great to advance science in the laboratory, but if you want to change the way patients are treated, you have to be able to do it in living systems. Ultimately, you have to be able to do it in a patient. I'm a physician scientist by training. I've spent my entire career looking for ways to take cutting edge science and apply it to patient care. And I've done that across large organizations, doing everything from discovery through clinical development on. Been fortunate to be part of bringing a large number of drugs to the market. And I just see enormous possibility in genetic medicine going forward. And in fact, by combining the delivery that we have at Tessera and the ability to correct errors in our genetic code or deliver new instructions through ultimately writing DNA into the genome, I think we can bring forward an enormous number of curative therapies in just the next handful of years. So gene writing is really a powerful tool in our hands, and it's core to what we do.
Good. Okay, thanks for that breakdown. It was really helpful for our listeners to understand what you're doing. And I'm really glad you brought up delivery specifically, which is where I want to go next. Delivery, is a challenge that we've covered on Cell and Gene. And in fact, I've moderated an entire Cell and Gene live event on the topic, because it's always been this sort of notorious challenge for gene editing technologies. And so I'd love to hear from you, when it comes to efficient delivery, how is gene writing different and why?
So gene writing is different in a number of ways. Importantly, the most advanced components of our gene writing platform are what we call RNA gene writers. These RNA gene writers are based on a family of mobile genetic elements called retrotransposons. And what retrotransposons do is they make copies of themselves and write themselves into DNA, but they do it through all RNA intermediates. So they're RNA templated, if you will. And they do that through a mechanism called target prime reverse transcription, which was first elucidated in the 1980s and 1990s. And so what we've done is harness that mechanism in our RNA gene writers to give us the ability to write permanent instructions into the DNA, but to do it with all RNA compositions. So the gene writer that does the work is a protein, but we can deliver it as a messenger RNA. And the template, which contains the instructions to be written into the genome, is also an RNA. And because they're all RNA compositions, they enable non-viral delivery systems. And those non-viral delivery systems have a number of advantages. So gene editing systems that require the delivery of DNA can't use non-viral systems today because the presence of foreign DNA in the cytosol looks to the cell like a virus, and it provokes a profound innate immune response, which is directly cytotoxic and also shuts down writing. But you can deliver messenger RNA and you can deliver template RNAs with those non-viral systems. So that allows us to use lipid nanoparticles. Lipid nanoparticles, in our view, have a number of very, very important advantages. First of all, they're redosable. They're very robust, highly scalable, and readily manufacturable. And we saw that in the experience with the mRNA COVID vaccines, where companies like Moderna and Pfizer and BioNTech scaled to the production of literally billions of doses around the world in a remarkably short period of time. But lipid nanoparticles have additional advantages in our hands because we've developed proprietary technology to allow us to essentially reprogram lipid nanoparticles to go to tissues that would otherwise be inaccessible. So lipid nanoparticles, when delivered systemically, naturally go to the liver through lipid and LDL receptor interactions. And we can deliver to the liver with very high efficiency. But we can reprogram those lipid nanoparticles to go to other tissues, to go to the resident stem cells in the bone marrow without mobilization, without chemotherapy conditioning, which opens up enormous possibilities like a true in vivo cure for sickle cell disease that doesn't require a stem cell transplant to administer. We can also reprogram those cells to go to immune cells, such as T cells, which allows us to pursue in vivo approaches for generating CAR T or other cancer fighting therapies and opens up an enormous set of possibilities as well. So by combining that RNA technology with our lipid nanoparticle delivery platform, we believe we can create a very large number of breakthrough drugs.
Gotcha. Okay. Before we move on specifically to Tessera and what you're developing specifically, talk to us a little bit about the differences between gene editing and gene writing.
Well, when people use the term gene editing today, they're typically talking about one of a handful of technologies. They're typically talking about either using CRISPR-Cas9 based systems in their first generation of approaches to knock genes out, as we've seen address certain conditions like the ex vivo stem cell therapy based, stem cell transplant based approaches to treating sickle cell disease and other conditions, or perhaps to chemical modifications of individual bases in the genome using similar systems. Gene writing is a much broader tool. Gene writing is a tool that gives us the versatility to make, as I said just a moment ago, virtually any change in the genome that's necessary to address human disease. So we can change any letter into any other at any position within the genome. We can write longer segments of DNA. So we can write whole exons and even longer segments into the genome with single nucleotide accuracy in terms of precision. And we can write whole genes into the genome. And we can do this with all RNA compositions. So that flexibility and the breadth of application is what differentiates gene writing from gene editing in our minds.
Gotcha. Okay, thank you. That helps to also set the framework for what I want to talk about next. So you're developing both RNA templated and DNA templated gene writing technologies based on engineered and synthetic mobile genetic elements, or MG. So talk us through a little bit, why both RNA and DNA, and then, maybe what are the advantages and opportunities for that?
So certainly the fundamental inspiration for our company was the biology of this family of genes called mobile genetic elements. They're evolutionarily ancient. We can trace them back literally billions of years through evolution, almost 3 billion years. And over that time, they've evolved various mechanisms to write themselves into genomes. And they're so successful at doing this that roughly half our DNA as humans comes from sequences that are derived from mobile genetic elements. So they truly made genomes modular, and we can think of them as core drivers of evolution and in a way as nature's genome architects. Now, there are many families of these mobile genetic elements, and we've studied them broadly. I described a moment ago our RNA gene writer platform, which is based on the biology of retrotransposons. We also explored DNA-based, DNA templated mobile genetic elements to form our DNA gene writer platform. The DNA gene writer platform may have certain advantages inserting very large segments of DNA. The RNA gene writer platform can insert up to whole genes, CAR cassettes, and similar length constructs. But if you want to get to 10 kilobases, 15 kilobases, as you might want to do for some certain applications, RNA gene writers may have an advantage in efficiency there. They also have some challenges today with respect to delivery because DNA gene writers require the delivery of DNA. And that can't be done through non-viral vectors, as I mentioned a moment ago. And the viral vectors all have in our minds, significant limitations. So the real potential of DNA gene writers, we believe, will be unlocked if we can actually overcome that hurdle. If we can find ways to safely and effectively deliver DNA through non-viral means. And I think that's possible in the near future, but it's not something that can be done today. So we continue to explore both, but the RNA gene writer platform is the more advanced of the two.
Sure, that makes sense. Thank you. Talk to us a little bit about what are the focus areas or unmet medical needs that you're targeting?
There's an enormous amount of unmet medical need that can be addressed by this platform. We're going to start off by pursuing therapies for either monogenetic diseases or cancer and autoimmunity. And we're going to do that because in those areas, the biology is very clear. In monogenetic diseases, we know exactly what the cause is, and we know if we can correct the cause, we can deliver a cure to patients as long as we do it in a timely fashion. And in cancer and autoimmunity, there's some very, very well-validated targets that can't be accessed efficiently with today's technologies. And that allows us to very clearly demonstrate the value of the platform, right? So we don't want to go into areas where the biology is unclear because then we won't know how to interpret the experiment, right? If we have a success, that's great. But if we don't, there'll always be questions about whether that was the right target or whether our technology performed in the way we wanted. By going into either those monogenetic diseases or those well-validated conditions, we can really isolate the impact of our platform and demonstrate its value very early on, and also bring forward some drugs that will make an enormous impact on the way we treat patients. So if you think about that spectrum of activity, in monogenetic diseases, there are conditions in the liver that have significant unmet medical need, are debilitating, life-shortening, and can be addressed with our technology. And I think a great example of that is a condition called alpha-1 antitrypsin deficiency. I'm sure you may be familiar with that condition, but a lot of people aren't, despite how common it is. And it's a disease that's caused by a single mutation in essentially all patients. And there's a fair number of these patients, between about 200 and 250,000 in the US and Europe alone, and there are other pockets around the world. And these patients have a mutation that caused them to form an abnormal alpha-1 antitrypsin protein that sticks to itself. So normally, alpha-1 antitrypsin is a protein that's made in the liver and it's secreted into circulation so it can go to the lungs and do its job. And its job is to act as a protease inhibitor. So what it does is it protects the lung from destruction by our immune systems as we go through the course of normal, routine immunosurveillance every day of our lives. So patients who have this mutation make an abnormal form of the protein that sticks to itself. It forms aggregates. And because it sticks to itself, it doesn't get out of the liver and it's directly toxic to the liver. And about a third of these patients will go on to have significant hepatic dysfunction, including cirrhosis. The second hallmark of the disease is because the protein doesn't get out of the liver and can't go to the lung to do its job, they get a progressive, destructive lung disease that's debilitating and life-shortening, and looks like a very aggressive form of chronic obstructive pulmonary disease, but it can't be managed well with existing therapies. And so by delivering our gene writer constructs through an LNP, we've shown the ability to correct that mutation with very high efficiency in humanized mice, and also in data that we presented at a meeting over the summer in non-human primates, writing it back to its intended sequence, to wild type. That allows that protein to do its normal job. It escapes the liver. The liver damage that shows up on histology in these humanized mouse models begins to reverse, and normal levels of circulating protein begin to be produced. So if we can do that in a patient, and we're moving rapidly towards the clinic, we can deliver a cure for a therapy for a condition that would otherwise have very, very limited therapy. And so that's just one example of what we can do with our technology in one of the areas that we're focusing on, which is the liver. But there's also enormous opportunity in other areas. I talked to you about stem cell delivery a little while ago. So we have developed lipid nanoparticles that can access the resident stem cell. So the true self-perpetuating, self-renewing stem cell in the bone marrow that produces our red and white blood cell systems. And we can take our gene writing technology and put it in that delivery system. And that allows us to do some remarkable things, like rewrite the hemoglobin locus. Ultimately, we intend to rewrite the hemoglobin locus in patients with sickle cell disease. And we're going to do this through a simple IV administration of a lipid nanoparticle gene writer construct. So I'm sure you're familiar with sickle cell disease broadly. One thing that not everyone realizes is that there's a significant burden of illness for sickle cell disease in the developed world. In the US and Europe, there are about 150,000 patients. And in other regions like the Middle East, there's probably about another 100,000 patients. There's 6 million patients globally, many of those living in underserved areas in equatorial Africa. And the therapeutic options for those patients are extraordinarily limited. There have recently been gene therapy approvals in the US and Europe at the end of last year, but each of those, as I mentioned earlier, requires a stem cell transplant to administer. That's not just a matter of convenience or delivery preference. These stem cell transplants require multiple rounds of stem cell mobilization, chemotherapy conditioning, so high-dose toxic chemotherapy, full-dose busulfan to completely eliminate their pre-existing marrow, which is then rescued with modified cells. Now, it's great to see any patient cured of sickle cell disease, but that's just not scalable. In fact, the large majority of sickle cell patients aren't even considered candidates for a stem cell transplant because they're too frail from their underlying illness. And even for those who are considered candidates, there are very significant health resource utilization limitations, just the number of transplants that the medical community can do, even in the US and in Europe, that will limit the broad applicability of those approaches. So by taking our gene writer, putting in that stem cell delivery vehicle that I described, we can convert that to a simple IV infusion, which we believe has the potential to rewrite that hemoglobin locus back to normal, so the normal adult hemoglobin. And we've shown in a number of scientific meetings that we can take CD34 cells from patients with sickle cell disease ex vivo. We're going to do this in vivo when we create a therapeutic, but we've started ex vivo to just demonstrate the value of the approach and get near complete correction and conversion to adult hemoglobin production. In a humanized mouse, we can put that in our delivery vehicle, and we can already rewrite the hemoglobin locus at levels that are above the anticipated curative threshold. And we're moving that forward into non-human primates, and we're moving rapidly towards the clinic. So we think that can really be a game changer for those 6 million patients living with sickle cell disease. And the opportunity to make that sort of impact is really what drives all of us at Tessera. And there are applications beyond that. I talked about cancer and autoimmunity. We can deliver instructions to T cells, either in a bedside, same-day process that eliminates the need for lymphodepletion, or through true just IV injection of lipid nanoparticles, so full in vivo production, to make functional CARs that can control cancers in xenograft models, and ultimately, we believe in patients, and can also be used to address autoimmune conditions. So there's just an enormous amount of opportunity that we can approach with this technology. And that's just the beginning. Those are the areas that I talked about in the beginning that have already validated biology. We can move into common disease, and we can move into many other areas over time. So the potential is really almost limitless, because there are thousands of conditions where we know our genes are the core drivers of both health and disease that can be addressed with this technology over time.
Yeah, sure. The potential is extremely exciting and fascinating. As I'm listening to you talk, I'm already kind of thinking about ways that, maybe down the road in the next year or so, how we can get you and your team back on to talk about the progress that's been made because it's just it's so exciting. But anyway, we'll talk about that at another time. What I want to talk about next is a press release from May of 2024 that I encourage our listeners to go search up. Notes that gene writers combined with proprietary lipid nanoparticle delivery to T cells allows same-day manufacturing with RNA-only composition to engineer CAR T cells that are cleared tumors in vivo in mice. And so I want to break that down a little bit because I want to talk about the in vivo aspect of that and then the same day manufacturing aspect of that. So manufacturing is a very, very important topic to Cell and Gene, the podcast listeners, and Cell and Gene readers. And so talk to us a little bit about not only the in vivo aspect of that whole thing, but also same day manufacturing.
Yeah. Certainly. So this applies to the T cell engineering programs that I mentioned earlier. And the press release that you're referring to refers to data that we presented at the ASGCT meeting in May in Baltimore. And so we have two complementary approaches for generating CAR Ts, both of which eliminate the need for offsite manufacturing and for lymphodepletion, which means chemotherapy conditioning, which is required with existing CAR T therapies today. The first of those is the process that you're talking about. It's a same-day bedside process. And what we do is we use a plasma, or rather a leukapheresis-like device to collect white cells at the bedside and mix them outside of the body with our LNP and then re-administer them. Okay. And the immune cells that have taken up the LNP then are transformed effectively into cancer-fighting CAR T cells in the body, and then they go about and do their job. And we've shown in xenograft models that a model of this process is very effective in controlling tumors. It can eliminate tumor volume and establish tumors in the way you'd expect an effective CAR response to do. So the advantages of that is that you have minimal manipulation of the T cells. So they're very fit. They produce very robust CARs. We would expect that to translate into very low doses required for an effective therapy and the effective establishment of memory, which is important for long-term outcomes and long-term tumor control, and ultimately for cures for some of these patients, as we've seen with the first generation of CAR therapies. So that approach has a number of advantages. One, we can combine the LNP with immune cells in very high concentrations because we're doing it outside of the body. If we want to multiplex, and our technology can be used to multiplex to make multiple changes, and if that multiplexing requires, in some cases, the administration of more than one LNP, we can do that in a manner that we know cells will be exposed to both LNPs. And it can be a very efficient way to create CARs without the need for that lymphodepletion. And so that's a technology that we think is very important. But we've also advanced the LNP technology to the point where we can go to a fully in vivo production. And by fully in vivo, what I mean is we can take our lipid nanoparticles, we can put our gene writing machinery in that lipid nanoparticle and deliver it in humanized mice through an IV infusion, and we can produce CARs fully in the body of mice. In this example, ultimately, we believe in patients. And that's a complementary way to get to that state of no offsite manufacturing, minimal manipulation, and no need for lymphodepletion. And so we think each of those will have its role. And so we view them as complementary technologies rather than, one or the other being the technology that we advance.
Thank you for that explanation. Before we wrap up, what's next for Tessera Therapeutics?
Well, there's a lot of exciting work ahead of us. I've been at Tessera for 2 years, and the amount of progress we've made in those 2 years is just remarkable. It's been, I think, one of the most productive, if not the most productive period of my entire career. I expect that to continue. We're going to continue to build out the data sets showing the in vivo potential of these therapies, not only in humanized mice, but increasingly in non-human primates. And essentially all of the aspects of our most advanced programs have now been demonstrated or will very soon be demonstrated in non-human primates. That's really important because non-human primate data are very, very predictive of clinical success in this space. I've spent 25 years doing drug discovery and development in large pharma. I've run large R&D organizations. I'm as skeptical as anybody about preclinical proof of concept. But it's different in genetic medicine. And the reason why it's different in genetic medicine is we're using these models in a very different way. If we want to make a mouse that looks like it has lupus, or looks like it has multiple sclerosis, or looks like it has some form of human cancer, and try to figure out how to treat that disease, we know it's going to lead us astray. But that's not what we're doing here. We know with absolute certainty what causes alpha-1 antitrypsin deficiency, what causes sickle cell disease, and so many of the other conditions that we're going to treat. So we're not using the models to figure out what causes a disease, and we know they're bad at that. We're using the models to determine, can we deliver our technology? And does our technology work in vivo in the way we would expect it to do based on all the development we've done up to this point? And when in primates, so a large animal closely related to a human, the answer to those questions is yes, then that translates to very high probability in the clinic. We've looked across the space of RNA therapeutics and genetic medicine, and in all reported outcomes, it's about 80% of the time that translates into the clinic. That's really important because you don't get to 80% probability that your efficacy signal is real in the rest of drug discovery and development until you've done a large phase 2 trial, a large mid-stage trial. In fact, most phase 2 trials have less predictive power than that. So these models are really important in this space, and we're going to continue to develop that data set. But in parallel with that, we're going to move quickly towards the clinic. We have a whole cohort of programs addressing significant unmet medical need in conditions like alpha-1 antitrypsin deficiencies we talked about, another genetic disease called phenylketonuria or PKU, sickle cell disease, and many others that we're going to be moving rapidly towards the clinic. So we're scaling the company and we're building the capabilities that we need to do that. And we'd expect to be in the clinic, in the near term. We're not giving exact guidance in terms of timing publicly, but given the progress we're making, I think you can see that it's not too far away. And then we really look forward to clinical proof of concept. So actually proving this technology in humans. And because of the nature of this technology, we can get that proof as early as the beginnings of the first in human study, the beginnings of the dose rising study. Because, for example, if a patient with sickle cell disease starts making normal hemoglobin, there's no placebo in the world that could make that happen. And we know that our technology is working in the way we like. And then it just becomes a matter of getting to the right dose and building the right evidence base. So I think there's some really exciting work ahead of us in the very near future.
All right, that's all for us on this episode of Cell and Gene, the podcast. Thanks to Tessera Therapeutics, Michael Severino, for joining me, and thank you for tuning in. Be sure to subscribe to Cell and Gene, the podcast, wherever you podcast, so you never miss an episode, and we'll talk to you soon.